I'd like to welcome everybody today to the February 22nd meeting of the IMAG MSM Working Group on Multiscale Modeling and Viral Pandemics. I'm very pleased to see you here. As always, I need to remind you that this meeting is live streamed on YouTube and recorded with minor editing for future viewing. Today we have Lourens Veen from the Netherlands eScience Center, who's going to be talking to us about multiscale integration. As always, Tom Helikar and myself are available to take suggestions and comments. We would also always thank Jim Sluka and Lorenzo, who are also available for your suggestions and ideas. Lorenzo is away today because of a family situation. We have our attempt at communication. Please do help us communicate. I think the speakers that we have deserve a bigger audience, and we have our wonderful archive of videos that really cover multiscale modeling in all sorts of different ways and deserve distribution. We are going to have next Friday a half-day virtual kickoff meeting for GLIMPRINT. It's going to be trying to develop a mission statement and initial purpose documents for GLIMPRINT. We hope you will come. The meeting invitation has been sent out by Lorenzo. Please do let us know if you're planning on coming. You're welcome to come without prior notice. You're not absolutely committed to come if you do say you're coming, but it would be helpful to have a rough estimate of how many people will join us to start trying to pull GLIMPRINT together. We would very much appreciate your contributions next week. We want to let people know about the Model of the Year competition that is being run by the group out of Biomodels in the UK. There is a deadline of February 29th for submission of models to Biomodels to compete for the most reproducible, high-quality reproducible models. As we all believe in model sharing and model reusability, it's critical that we make an effort and recognize people who make the effort, because it is an effort, to make models reusable. And Sharif would be happy to talk to you. Please remind your students, postdocs, if they have been publishing in a way that is in fact reproducible, please do consider competing for this award and contact Sharif. I don't see is Sharif on the call today? No, he's not. So I have to speak on his behalf. I need to remind people that Indiana University at the moment is conducting a search in the general area called biomedical engineering, bioengineering. We have 3 open positions this year in computational biological subjects, computational bioengineering. You can find the listings here. If you search for Indiana 22693, 22623, you'll find those announcements. I'm the chair of the search committee, so if you have any questions about that, please come to me. I think people with connections in this community might very well be people who would be appropriate for this search. I also have an announcement from my end. We have our Open VT community building exercise in multicellular modeling. We've contacted many of you individually. We are now trying to expand that community, decide what our activities in terms of stakeholders should be, what our stakeholders should be, what our activities should be, what services we should provide. And again, I'm happy to hear from people if you have suggestions. We will have a steering committee meeting tomorrow at 11 a.m. As usual. I believe that Tom will not be available, but if you believe you need an invite and you haven't received one already, please do feel free to connect to me in the chat if necessary today, and I'll be happy to send you an invitation for tomorrow. In particular, it'll be organizing that GLIMPRINT meeting for the following Friday. Are there any other announcements to make at this point before we move forward? Okay. So on March 7th, we have Belinda Akpa from Oak Ridge National Laboratory, who's going to be talking about a Molecules to Life modeling initiative, cross-scale modeling initiative. April 4th, we have Rebecca Morrison talking about model calibration, validation, uncertainty quantification for stochastic models, both topics I think of great interest to the community. We have an open slot on March 21st. Please do help us. As always, you've heard me say it many times. Feel free to suggest yourself, even if you've spoken before. If you have new work, new ideas to present, suggest your graduate students or postdocs to speak if you like. We're always glad to hear early career stage speakers. If there are people whose work you've admired and you'd like to hear from, please let us know and we'll get Lorenzo to do his best to try to get those people on the roster. So I don't want to take more time away from Laurens. We're going to be hearing about cross-scale, multi-scale simulation coupling and the work that he has been doing in this area. And I'll turn the screen share over to him.
Okay, good afternoon, or possibly evening, if there are any other people in Europe. My name is Lourens Veen. I'm a senior research software engineer at the Netherlands eScience Center. I thought I would just briefly introduce us because you probably haven't heard of us. We are the National Center of Expertise for Scientific Software. And perhaps it's easier to say what we're not. We're not an HPC center, so we don't run any supercomputers. We're not a research institute. We're not a university. We kind of operate in the area in between all of those things. So we work with scientists. We are big on collaborative science. And we work with scientists who need or use software and want help with that. We also cross all disciplines, so we work from archaeology to astrobiology and from evolution to law, really anything, including humanities and social sciences. And we mostly focus on methods and tools. So we work with scientists on their research, but then help them with methodology, with making software, with using software, and sometimes accessing computer resources, all these kinds of technical nuts and bolts of doing science. So that also means that we're not exclusively doing computational science. It's not all simulations. It's also lots of data processing and really all sorts of things. It's a very interesting place to work. Briefly about myself, just to help communicate a little bit, because I understand this is a multidisciplinary group. I'm a computer scientist by training, ended up in biogeography, did that for a couple of years, and then I've joined the eScience Center where I've worked in a variety of fields, but they've all sort of been computational projects. And so I'm more and more considering myself a computational scientist, although I kind of feel like I don't understand enough physics yet to really be able to call myself that. So somewhere in between. I'm going to talk about coupled simulations, but just to kind of introduce the topic and explain why, at least why I think it's important. We seem to have a lot of large challenges in this century as a species. And these challenges are increasingly, scientists are being asked to explain how complex systems work. And not only that, but also to give advice on how to steer them towards a better future. And so this is posing all sorts of challenges, not just because of the complexity of these systems and because of the emergent behavior that they exhibit, but also almost from a science philosophical perspective. We don't have a second Earth, so we can't do a reproducible controlled experiment because we have no control. And also when you start modeling these complex systems, you often do that at a higher level of abstraction. And that means that you're going to introduce concepts that aren't directly measurable out in the real world. So I think we need computational science to fill that gap, and we need simulations to try to predict what will happen if we can't do an experiment. And there's a bunch more things to be said about that, but I need to get on to coupled simulations. So yeah, I think Rebecca Morrison is going to present on uncertainty quantification and various related things. I've also done some work in that direction, and I think there's a lot of connections with that here. At any rate, computational science, also digital twins, seems to be on the rise, and I think that's good news, at least for us. So when you want to design such a system, it's just to kind of summarize what I'm going to talk about. This is a brief introduction. After that, I will talk about theory for coupling and a bit of practical implementation using the software I've developed. If you're going to model a complex system, there are actually several challenges that you need to overcome. And one that I've been thinking about more and more lately is the conceptual design. How do you go from all sorts of possibly very detailed knowledge about various aspects of this system to some kind of overall design of how you're going to make a model? Which parts does it consist of? Which submodels are you going to have? And the reason I'm thinking about this is that many of the people I work with seem to be struggling with that part. And it's interesting, my background in computer science is in information systems, which are things like your personnel administration. Well, you can see why I went into computational science instead. But those systems also describe the real world. And when you design one, you need to think about that real world and you need to model it somehow. And that's not an easy thing to do. But there are some techniques from that field that I think might apply in computational science, and it's something I would like to explore more in the future. For now, I have a question there. I don't have an answer. It's a very nice place to be, but it does mean that I can't present you that answer today. So instead, I'm going to talk more about what happens after that, when you know which submodels you have, and about the semantics of the interactions, and about the technical construction of the model. So the first part of this presentation is about the multi-skill modeling and simulation framework. This is a theory of coupled simulations that was developed already a while ago at the University of Amsterdam and at the University of Geneva. So they work together on this. Professor Alfons Hoekstra at the Computational Science Lab of the University of Amsterdam has been leading this. And his PhD student, Joris Borgdorf, he has been instrumental in deriving a lot of the theory. And so what this is a formalism for multi-skill model coupling. And then there's Muscle 3, which is software that implements that formalism and helps you actually make that work in practice. Now, I became involved in this when Alfons wrote a proposal for a collaboration with us at the eScience Center. And this proposal was about uncertainty quantification of coupled simulations and taking advantage of the fact that it is a coupled simulation to do UQ more efficiently, for example, by using different ensemble sizes for different submodels. Like I said, I'm not going to go into that here because one thing at a time, but maybe in the future. I'm looking forward to that presentation in April at least. So because abstraction is always more difficult to explain than a concrete case, here's a concrete case study. This is a model that Professor Hoekstra has been working on with various people. It's a model of instant restenosis. This is an adverse treatment outcome of balloon angioplasty and stenting after atherosclerosis. So in plain English, what that means is atherosclerosis is the narrowing of the coronary artery with a fatty plaque. This is a very common ailment. And you can treat that by inserting a balloon inside of the artery and expanding it, which flattens the plaque against the site so that the blood can flow again. And then typically a stent is inserted. You can see that at the bottom here, it's this kind of wire mesh device that holds the artery open. Now, this does damage the artery a little bit. And in some cases, so you get a healing response, but in some cases that healing response is too much of a healing response. And so the cells that are supposed to heal the artery keep growing and the whole thing closes up again. And that's obviously not good. You can get a heart attack. So they're trying to simulate this to see if we can maybe change the stent design or if there are particular patients that are more at risk. And this model consists of a number of submodels, 3 in fact, at least in this slightly simplified representation. The first model is at the bottom left. This is the model that calculates the initial condition. So effectively, it simulates the stent deployment. And then at the top, we have the smooth muscle cell growth model. So this models the cells in the wall of the artery. And then we have a blood flow model, which is a computational fluid dynamics model that models the flow of the blood through the lumen, which is the open area in the middle. And these processes actually influence each other. So the cells can sense the flow of the blood, and that can inhibit their growth at some point. But also, of course, the growing cells change the geometry of the blood vessel and therefore change the way that the blood flows. So this is a coupled simulation. And if you want to think about how to couple these models, the first thing you need to know is actually what is a model. Now, in the MMSF, this is a model. And what this diagram means is that every model starts with an initialization phase, during which you can get some input from the outside world. So commonly you load some data from disk and initialize your state. And then you're going to write some output or produce some output somehow. Usually you write an image to disk so you can make a nice video later. Once that's done, you compute the state update. So you compute the next state from the current state. Almost all simulation models actually time step. Not all of them, but if your model is a simple function, what you can do is just do this loop, because that's what it is, 0 times, and then you have a function, so it fits as well. So you calculate your state update. That gives you your state at time t is 1, and you produce another output, and you can repeat that for however many steps you want until you reach convergence, or until all the interesting dynamics are done, or until you get bored or run out of compute hours. So finally, you reach some kind of endpoint, and at that point, you're going to have a final state, and usually from that final state also some output is produced. These kinds of blocks, loops, these are the fundamental building blocks of a simulation in the MMSF and of a simulation in muscle. And if you've ever written a simulation model, then you'll probably recognize this. So then the question is, how do you connect them together? And broadly speaking, there are 3 different ways of doing that are out there. Usually they follow one of these models. The first one, and probably the most popular one, is what I call scripting. In this case, you take your model, written in whichever language it is, and you convert it from a program that runs a simulation into a library, usually with a Python interface. And then you write a Python script that calls all the different models in a particular order and runs them for a bit and extracts some information and sends it to another one. Systems like FC2K, which is a bit older and I think fairly widespread. IRAP is used in the fusion community. Amuse physics, the basic model interface is geophysics. All these things work in more or less the same way. Then there's a second kind of system, which is a workflow management system. These were quite popular some years ago. These build models or data processing systems also, because that's what they're commonly used for, out of functions. And they build a directed acyclic graph of functions. And that's often problematic because you want to have a loop somewhere. Some of these systems like Kepler support this and then have been used for model coupling as well. And then finally, there's a paradigm which I'm calling actors here, which for the computer scientists is not entirely accurate because the scripting thing also has actors technically. But what I mean here by this is that your simulation consists of a number of programs, independent computer programs that are all running simultaneously and that exchange data with each other while they are running. This is done with MPI, I think, in the climate community. This is also how the MMSF and how muscle work. And at first glance, that may sound very complicated because it's always more difficult to have more than one program and have to think about all of them simultaneously. On the other hand, keep in mind that we're modeling complex systems. So your model is going to be complex. It's going to have a lot of moving pieces and they're going to interact in all sorts of ways. By definition, that's what the system does. So if you take a scripting approach, it may sound very easy because everything is in a single script, but what you're really doing is you're taking all that complexity and you're just sweeping it into a single box. And then you have to go and actually implement that box and you find yourself faced with a very complex programming problem. So I think at some point your system is going to get complex enough that it's actually better to split it into separate programs. And this is often also convenient because you can reuse some of those programs. Like the computational fluid dynamics code, that's a standard off-the-shelf code in the example I gave before. So I think ultimately that this works, but the way to couple it can be a bit tricky. So to get back to this example, hold on. Yeah, so we have 3 different programs actually, one for the initial conditions, one for the smooth muscle cells, and one for the blood flow. And the next question is then, how do we couple these things together? And that's shown in these diagrams. What you see on the left here is the domains of the different processes. So we have the stent deployment, which takes place in the wall, like the stent expands and that affects the cells, but it doesn't affect the flow of the blood, at least not in the simulation. And anyway, it's near instantaneous. And then also in the wall, there's the cell model. So the cell growth takes place in the arterial wall. And then in the lumen, there's the blood flow. So these domains are adjacent in space, but we also have domains that are adjacent in time. The stent deployment happens just before the rest of the simulation. On the right, we have a scale separation map. So this shows how large and how fast the different processes are. I've removed the stent deployment here, so it's just the blood flow and the cell model. And what we can see here is that in space, on the vertical axis, these things are about the same size, which makes sense, right? They model the same section of artery, so that's about the same overall size. Also, in terms of the resolution, which is at the bottom of the boxes, the individual smooth muscle cells are about the same size as the grid spacing of the blood flow model. So in terms of the spatial scales, they are about the same. In time, however, there's a scale separation because the blood flow is much faster than the cell model. These cells take something like 26 hours to grow and mitose or split. And blood flow, as happens with your heartbeat, so we're talking less than a second. The cell model has a time step of about an hour. Blood flow is much smaller. So we have a temporal scale separation here in this coupling. Now, if we apply the theory to this example, then what it says is that because the stent deployment goes before everything else, we should take the final state of the stent deployment process and use that to inform the initialization of, in this case, the cell model, because they're on the same domain. They're both in the wall. And then between the cell model and the blood flow model, we are going to have a coupling where the cell model effectively calls the blood flow model for every time step. So we get the cell model, and then we, at every time step of the cell model, we're going to do a full run of the blood flow model because it's much faster. So this is a typical multi-scale coupling. And it can be implemented by taking the 2 operations in the loop, so the state update and which you can see at the top right, the state update in the intermediate output, and using that to connect to the initialization and the final state of the blood flow model. And what you get then is what you see on the right, where the stent deployment runs first, and then it starts both of the models actually, but we get a full run of the blood flow model for every time step of the cell model. Now, there are other coupling patterns that you can get depending on the situation. So this is one example. But overall, to summarize the theory, if you know your domains in time and you know your scales in time, and you know your scales in space and your domains in space, then you can decide which of these couplings, there are 3 possible variants here, you use. So we've seen dispatch, that's if something happens before something else. Call and release is for temporal scale separation, and interact is for temporal scale overlap. And then you can decide if your domains are adjacent, you're exchanging boundary conditions, but you can also, for example, in a reaction diffusion model, have a case where 2 processes take place on the same domain. And in that case, you need to exchange the state if you want to implement that as separate programs. And then there's one more thing. If you have spatial scale separation, so one process is much larger than the other, what you tend to get is a lot of instances of the small model, for example, for each grid cell of the large model. So in that case, you need to have multiple instances of a particular model and a way of communicating with them. And that is also something that the NMFSF models and that Muscle 3 provides. So this is in theory. In practice, this is all nice, but you still need to go and connect your scripts or codes together. And for that, we have software. Muscle 3, which is the most recent incarnation of the multi-skill coupling library and environment. Not to be confused with the other muscle, which is a sequence aligning tool that's used a lot in various kinds of omics. They started around the same time and picked the same name, unfortunately. So sometimes I get a help request from someone for software that I don't actually know anything about, but mostly we get by. So a muscle 3 simulation looks like this. We have 3 programs at the bottom. Those are basically the normal simulation codes, except that they link with an extra package or a library, which is called libmuscle. And then there's the muscle manager, which is in charge of controlling the whole thing, which reads a configuration file at the top left called the YMMSL file. And how this works is that this configuration file actually describes which submodels there are and how they should be connected. So that is not hard-coded in the code. And that means that it's very easy to actually change which models you have and how they are connected. This also contains some settings and things like how many resources, how many CPU cores to give to each model and those sorts of things, how to start them. So the manager reads all this, and then it's going to decide where to start the models. If you're running on your laptop, that's just all on your laptop. But if you're running on an HPC machine, then you have an allocation with multiple nodes, and it needs to put each model somewhere. It can actually analyze the coupling pattern and decide that, for example, since stent deployment happens before everything else, it can share the same course because they're not active at the same time anyway. So this can save you a lot of compute resources and save us a lot of energy as well. Once it's decided where to put everything, it starts up all the programs. The programs start up a network server. And then they connect back to the manager and say, hey, I'm here. Who should I talk to? And the manager tells them that. And then they set up network connections directly, which are used for the communication. So it's not communicating via disk. It's going via the network, which is faster. And it's also going via the network directly between the models, which is really nice because if you add more models, you can also add more compute resources and with that add more network cables, and it just keeps scaling. So once everything is running, the manager is just collecting a bit of logging information and some profiling data, but it's not really involved in the execution. So it's not like your Python script where the Python script actually drives everything. The manager is just there to facilitate. This I already mentioned, so this is in the configuration file. There's a central setting system, so you can set one setting that all the models can pick up, but you can also set them specifically for individual instances. So it's a YAML file, the configuration. This is what it looks like. So basically on the left, what you see here is a textual description of the diagram that you see at the top right. It's really quite simple. So I have another example. This is work I've recently done with Professor Roland Merks at Leiden University. He's interested in embryology and tumors. He's a mathematician and a biologist, and studies interactions between cells, interactions between endothelial cells and the extracellular matrix, which is the sort of fibrous matter that they're inside of in your body. And the thing is that these cells can actually pull on this to move around, but also to signal, to sense. They can secrete substances that work as signals for other cells. And so what he's interested in is how you get from a single cell that then starts to divide to cells that team up together and form capillaries and later arteries and larger structures. So what you see on the right here is one of his models. This is from a test run, I think, so this is not a realistic situation. But what you see in the center, the red dot is a cell, which is simulated using the cellular POTS model, which is familiar to some of you. And in the background, you see a coarse-grained molecular dynamics simulation where we simulate the extracellular matrix. And then if you look carefully, you can see some dots. Those are so-called adhesions, which is sort of little grippers that the cell uses to grab onto the ECM. So I went and recoupled this model using Muscle 3, and I've made a diagram here. I'm not going into too much detail. What you can see is that it's actually somewhat similar to the instant restenosis model that I showed before. We have some initialization phase where we make the extracellular matrix, produce all the fibers, and that needs to be equilibrated because there are some internal forces. And finally, we have two processes working simultaneously, one to simulate the extracellular matrix and one to run the cellular POTS model. Timescales here are actually a bit difficult to model. I'm still not entirely sure whether what I've done here is theoretically correct, but it works in practice. And I think that's also okay, right? Both the MMSF and Muscle3 are tools. It's not a religion. So as long as your scientific results are correct, and it works for you, then I'm perfectly happy too. One extra thing that we have here is this thing called the state viewer. This is an extra component that isn't really part of the model, but it's just a technical component. Every once in a while, every n time steps, basically, the simulation, the ECM simulation and the cellular POTS model are going to send their entire state to this component, and it will plot it to the screen. If you're running locally, if you're on HPC, what you do is you put a state dumper in there that saves it to disk so that you can later make a nice video. The models here on the right, they don't do this call and release thing that the instant restenosis model does, because I've modeled it with a temporal scale overlap. So instead, they run in lockstep, which means that they both do, so they both start at TS0 and then they both step to TS1 and so on, exchanging information at every step. So you can see that here. This is actually a timeline plot that is produced by Muscle. So it collects this information automatically. You don't have to do anything for it. Once your run is done, you can open that file with the little tool that's included, and it will show you some information about how it's been running. So what you can see here is that at the bottom left, we were making the extracellular matrix, and then we were done. It got equilibrated in the second lane from the bottom. And then there was some initialization going on because the models need to make the adhesions. So that's a big block there in the simulate ECM column, the second from the top. And once we get sort of to the middle of this plot, you see that the cellular POTS model and the simulate ECM model start running at the same time. But the simulate ECM model is faster. So it runs for a short time, and then it has to wait for the cellular POTS model to be done and send it the boundary conditions that it needs to continue. In terms of the resources, Muscle is going to put the make ECM and equilibrate ECM on the same nodes as simulate ECM because they don't overlap, but the ECM simulation and the cellular POTS model are actually going to be on different nodes or different CPU cores, or in this case on a GPU and a CPU, because they are active at the same time. So they can't share resources, and Muscle will determine that automatically and take care of it behind the scenes, so you don't have to worry about it, but you do get the benefits. So briefly, features, Muscle does multi-scale and also multi-physics coupling. It's designed for multi-scale, but it does do multi-physics as well. It's got APIs in Python, in Fortran, and in C++, and we're looking to add a couple more languages like Julia, maybe MATLAB, and any other language really. It's quite modular, so we can just add bindings for other languages as we need. It runs locally, it runs on HPC. It comes with a bunch of examples and some quite extensive documentation. Actually, there's so much documentation now that I'm planning to redesign it a little bit because it's grown quite a lot. And finally, it's open source under the Apache license and it's developed openly on GitHub. So you can start using it right now if you want, no need to ask. I've shown two applications in sort of the biomedical domain, but I'm working with other fields as well. So I have a simulation, or rather Professor Peter Coveney in London at University College London has a simulation of the human circulatory system. So that's what they're interested in. And that is now running with Muscle. And they also have an application in advanced polymers and crack propagation, which is sort of the traditional multi-scale problem that they're looking to Muscleify. And another big interest and working domain of mine over the last couple of years is to work with the fusion community. In tokamak modeling, those machines are very complicated. There's lots of stuff going on across scales. And also they have a control system that they're interested in simulating and integrating into the simulation. And recently I've been contacted by some people who are doing electrophysiology. So we'll see what will come of that, but it's still very early days. So I'm running out of time. To summarize, I think coupled simulations are needed to answer questions about large complex systems, and we have more and more of those questions. Fortunately, we have the multi-scale modeling and simulation framework, which tells us, at least in terms of, in somewhat abstract terms, because it doesn't solve the physics or the biology of the couplings, but it does tell us how to deal with time and space and domains and scales. And it gives us these coupling templates that show how to do it. And then we have the Muscle 3 software that implements all of that and puts the model structure in the configuration, so you can have multiple versions of your model that you can easily compile, easily compare without having to maintain multiple branches of your model code. It works across languages, so they're also interoperable. If you send an array in Fortran, you will receive the corresponding NumPy array in Python, if the receiver happens to be written in Python. And it's open source and it's easy to use. So if you're doing something like this, then maybe you should give it a try. That's what I had to say. Looking forward to questions and the discussion. Thank you.
Okay, thank you very much. The floor is open for questions.
Yeah, I've got a question, I guess. This is Randy Highland. Just jumping right into it, is it possible then to simply download and run locally Muscle with one of your examples, one of the examples that's included with Muscle, if I am on, say, a Windows laptop?
Yes, but on the Windows subsystem for Linux. Actually, I think the next version, as long as you're using only Python, might work on Windows plain as well. The C++ and the Fortran code hasn't been tried on Windows. So I would not expect that to work out of the box. Usually there would be some small things needed.
And what about Mac?
It does work on Mac, yes.
Okay.
Other questions? For example, I mean, Reinhard has worked extensively on a rather different approach to model, multi-model integration, more on the data side. I know. Maybe it would be worth thinking about the different philosophies of model integration that those represent.
I just sent Lourens a copy of the papers. Essentially, the idea was that the way we do modularity is we decompose the simulation model rather than the mathematical model, and that enables you to not have the different components interact directly. They interact through modification of the data space.
Okay, so you get, this just reminds me a little bit of a coupling system called Precise, which I think is German and a little bit Dutch. I think they do a similar thing. So they define basically global variables that you can read to or write from, and the different models do that. And that is how it's sort of a data-centric architecture, I would say, as a computer scientist. Is that about the right understanding?
Yes, I'm not familiar with this particular approach, but if you have a chance and you can take a look at my paper, I'd love to get some feedback from you.
I will have a look. Yes, it sounds interesting. Did you send this to me by email or?
Yeah, I think I sent it to your University of Leiden email address.
Oh, okay. I will, yeah. Go and find it there then.
Yeah, I just looked you up on the web.
Okay. Oh, so that's interesting. It's yeah. So I'm a guest there actually, but I read that email too. So.
Okay. Well, if you don't get it, let me know.
Yeah, I will.
Other comments or questions?
Yeah. So
go.
I was going to say I can ask a question.
No, please go ahead, Mitchell.
Yeah. So this was a fantastic talk. I think a lot of us, especially, so I'm in cardiovascular, so a lot of us tend to think about growth and remodeling, sort of the cell response to various mechanical stimuli, like we've gotten the fluid and solid mechanics down, how we got to really integrate across scales. I don't think you mentioned this, but when it comes to looking at things like, if you're going to do an inverse problem, or even if you're going to do uncertainty quantification, I can imagine across scales, right? There might be some relationships that are sort of maybe not intuitive at a first glance, but maybe once you start doing some forward evaluations, you start to notice this. Are you looking at sort of inverse problems from a multi-scale perspective and how you can maybe integrate that into your framework? Or in the same question with sort of UQ, are you looking at mostly non-intrusive UQ methods? Or are you looking to do a little bit of a hybrid and things like that? I'm sort of curious with this software, what your approach is going to be.
Yeah, thank you. That's an interesting question. So in the project that I did with Alfons Hoekstra and his PhD student Anna Nikishova, maybe that's a name you should look up. We looked at forward UQ, And in combination with surrogate modeling, and also with changing the sample size per submodel. And what we actually developed there, or what Anna developed, is something that we call a semi-intrusive method. So you can have fully intrusive methods where you essentially change the model equations to propagate not a single state, but an entire distribution, usually using some form of polynomial chaos expansion or something to keep that manageable. And then there are non-intrusive methods like Monte Carlo and various versions of that, where you just calculate a whole bunch of samples. What we've done here is what we call a semi-intrusive method, is a method that leaves. So that the advantage of a black box of a Monte Carlo method is that it considers the model a closed box, so you don't have to change your model. What we've done here is what we call semi-intrusive, is where we change the way that the different submodels are connected together, and maybe how many instances of them there are, maybe we put some additional things in there, but we leave the individual submodels as closed boxes. Or perhaps if you wanted to, and you had an intrusive version of one of them, you could substitute that in and combine it with a sampler that surrounds another submodel. So you can connect them together in some way. There are some potential statistical issues that you get in these kinds of couplings because you get problems with covariances. But not always, things are not always correlated. So if they aren't, then that isn't an issue. So we've done that. And one of the interesting things you can do with muscle is if you have an ensemble, you have all these programs active at the same time. And so you can start doing things like taking data from different universes in your multiverse, so to speak, and actually use that to train a surrogate model, which you can then substitute to run your UQ faster. And we think it's even possible to do this on the fly. And we've written a paper about how to do it in theory, but we didn't get around to actually doing it in practice. So if you look up that name, I'll just write it in the chat. She has written a thesis about it and a series of papers. So that will put you in the right direction there. As for inverse uncertainty quantification, we haven't done that with muscle yet. I have done it in the past. And I think that inverse is where you start doing parameter estimation and that sort of thing. So this is where the no second Earth problem comes in. That is also something that you can do with muscle. The problem with that is, of course, that it's often expensive. So I've worked with Markov chain Monte Carlo methods, and you need to do a whole lot of model runs. I think there are better ways of doing it that I'm not so familiar with. But it's something that for sure you could do, because essentially you're just repeatedly running the model in all these cases and sometimes taking information. So in muscle, how you couple a macro-micro model is that essentially the macro model couples the micro model. I showed that for the instant restenosis model. You can use that exact same pattern to have a sampler that runs a simulation many times and perhaps resamples something or uses information in between, or runs a whole bunch of those models in parallel. So the same thing, the same multiple instance mechanism you use for UQ, but also for spatial scale separation, you can also use that here.
Very nice. And then, so thank you for that. And I might have missed it too. Did you say in the software, let's say I have a very, I want to take a different approach for smooth muscle cell contraction. And so I want the coupling to be the same, but my micro scale needs to be adapted. Is it possible in this framework to sort of go in and say, well, everything should be fine, but what I believe about the micro scale should be adapted to this model type, but keep the coupling the same? Is that feasible?
Yes, so you can take one of those. So one thing that muscle does is it takes multiple configuration files and it just glues them together. So you can override something by adding another model on. So for example, in the cellular POTS model and an ECM simulation, that state dumper and the state viewer are actually in separate configuration files. So they just get added on if you want them. But you can also just copy paste your configuration file and then add the simulation there. So if you want to substitute a different model or a surrogate model or something, you need to implement that. And then you just tell muscle how to start that program instead. And you put it in there, you tell it to use that instead of the others, and then rerun the simulation. And hopefully get exactly the same answer or a better answer, or well, something that you can compare and publish about at least.
Perfect. Thank you.
I think this is actually particularly nice because the In many cases, if you have a coupled simulation, if you do it, for example, with MPI, then the coupling itself is kind of hard-coded in the models. So if you want to change something, or you want to put some kind of a cache or a buffer or some other kind of technical component in between, you have to actually modify the code. And then if you want to compare things, you need to start maintaining multiple versions, and then you find a bug and you fix it on one branch, but not on the other, and it turns into a big mess. So it's actually very nice to just be able to do this completely separately. It depends a little bit on how much effort you put into coupling your models. So you can make a really nice generically reusable component that can take all sorts of inputs and produce all sorts of outputs and is very configurable. That takes more effort. If you have a particular fixed combination of models that you want to run, you can also just do it quick and dirty, and then you can only couple them in that way. But maybe you don't even know if it's going to work, right? So you don't want to spend too much time. I think it's important to have that option.
Other questions?
Are there many people here who do coupled simulations? And if so, how do you do this?
Well,
Usually
right.
by hand rolling code. That's what we've done in the past. Linking, a whole body PBK model to a blood flow through a pipe or something like that, and just lots of custom Python code to make the two models work together.
And is
Yes,
that... Go ahead.
sorry. Yeah, same for me. I model the transport of particles in the lung, and so we have the multi-scale come from the model that simulate region of the lung, going from full CFD in the first generation and going to a 1D model in the deeper lung. And so you have several outlets, and we do a script with Python of the output of Fluent that takes, or whatever CFD code you use, that's more complex output, and we transform it to go into our 1D models, which run in a few seconds. So we kind of do it not simultaneously because the 1D portion is so fast that there is no need for us at this stage. Yeah.
As we say in computer science, premature optimization is the root of all evil. If it's fast enough, don't touch it.
Exactly.
Oh, that's very good. So it's...
I mean, I think we also...
using some kind of coupling framework, or?
Sorry, Botchek and TJ should probably speak up, but we typically also basically have a Python scripted interaction between our solvers. Something that I'm always thinking about when I hear talks like this is, what level do you call a submodel? Right? I mean, you could repetitive, right? Even the individual solvers actually have subcomponents to them, and so one could potentially iteratively do this a number of times. There are certain things that are clearly sort of atomic solvers which you don't want to break up. But if the architecture is efficient enough, you'd gain by doing that breakup. Classically, the worry about doing this kind of approach was that if you had tight coupling, the communication overhead between the processes would kill you. And the example you give of Lourens is one where I would have expected that would happen, because the coupling between the fibers and the cell is happening in the innermost loop of the solver for the pot solver. In other words, the fibers are applying a force, and that force is being evaluated in the innermost loop of the POTS algorithm. And so to me, the coupling between the fiber architecture and the cell movement is, in a sense, as intimate, as tightly coupled as I could almost imagine a process to be. There isn't a separation of time scales. There's no separation of length scales. It's a really hard problem. Well, you might have to solve the fibers reorient faster than the cells, so you have some time. You have to solve the fiber movement faster even than that. But it's still it's a tough problem, and I would have thought potentially the overhead of having the communication might be so expensive that you lose the benefit of the clarity of the architecture. Clearly, that's not the case. Not saying that I'm impressed, but it does ask the question: how do you decide what level of decomposition to use?
Yeah, I have to confess here, because it doesn't quite work that way. You're correct, of course, because you invented the thing that the cellular pods model would have to, when it tries to do a copy attempt, it needs to move the adhesion along, and it needs to evaluate the forces between that adhesion and the ECM fibers that it's attached to. What I actually do is I'm sending on every macro time step of the cellular pods model, so every Monte Carlo step, I'm sending the positions of the fiber particles that are attached to the adhesions from the ECM to the cellular pods model, and within this macro time step, they don't move. So I am evaluating the forces at every micro time step of the CPM, but I'm not updating the information because it does millions per second, and not even with MPI or shared memory, there's just nothing that's fast enough to do that. So essentially the locations get cached, and then at the end of the time step, the cellular pods model, it records all the movement and changes to the adhesions it makes. So those move. And those changes are also sent to the ECM, which then re-equilibrates the fibers. So the models are essentially diverging a little bit because they're both using the old state of the previous one, and then they get resynchronized, so to speak.
So that's the typical way we do it, which is just a standard time slicing algorithm where you freeze everything that's not inside your solver during the time slicing.
Yeah.
The usual cost of that is that the maximal time step for what you call the Monte Carlo step, that has to be in a very short amount of time, because you can't have a significant displacement of the other system, because if your parameters get too stale, everything becomes unstable.
Yeah, no, that's correct. I don't really have a better solution for this either, so that's I did it the usual way, I guess.
No, that's fine. But I appreciate that you're working on an intrinsically very hard problem there.
Yeah.
I think that the stenosis one perhaps is, despite perhaps being numerically heavier, it may be a little bit easier because you really have such a clear separation of timescales, where the fluid dynamics happens on such a fast timescale compared to everything else.
Yeah.
That you really can do a separation of timescales problem.
Yeah, that's a very nice mechanism, and it really helps there as well. The blood flow simulation there is still the most expensive part of the model, and this is actually a fairly simplistic flow. So it's essentially thick water. There are simulations that actually simulate the red blood cells in the blood, and you get all these interesting shear thickening phenomena. And actually, at the University of Amsterdam, they do have such a model as well. It's just not used in the simulation. But even then, it's already quite expensive. So I think in terms of communication performance and latency, Muscle 3 has a minimum latency of about 250 microseconds at the moment, which compared to MPI is actually not so good. I can do some optimizations there. So I would like to actually make it faster. The problem is that all my users keep telling me, no, it's easily fast enough. Don't worry. We have other things we would like you to do first. So it's quite interesting that apparently, I think people do sort of more the large scale couplings, and if they have something very tightly coupled, they just put it into a single program, and then you don't have this issue. Probably the way to go in many cases.
Well, the community that we've been talking to, my introductory remarks about this sort of general open VT concept, there does seem to be a growing desire to have some kind of back end, I would say API, but that's really too limiting. Some way of interchanging solvers that would work. And you're potentially offering us a way of doing that kind of interchange, where if somebody wrote a really good PDE solver, provided that we were compliant with Muscle 3, we could then, and they were compliant with Muscle 3, we could then potentially use that tool.
Yep.
I think that even if there were some computational penalty associated with it. I don't think it sounds like that's heavy. The flexibility and the ability to disseminate best practice in solvers by itself would be a huge advance in our field. I don't know about others. I know that Mitchell, you talk in cardiovascular, you're more sophisticated than we are, I think, in the multi-cell community, perhaps. But I'd love to hear what other people's experiences are about that, if people have feelings on that.
I was going to say, so in cardiovascular, like I think everyone's moving to multi-scale, but how you do it, I think is very much, I think this is the reproducibility issue is going to get worse because everyone's doing it their own way. Yeah, I wish I had a good answer for that. I mean, I think the software is very relevant. I think the other thing too is so many people now are doing surrogate modeling, and a surrogate is also a very, individual dependent idea. If I want to use a Gaussian process, I want to use chaos, I just want polynomial. Like, I feel like that issue is going to come up, especially as we talk about digital twins, because no one's going to run their full simulation. Yeah, I don't know. And then, I can imagine for your software too, you're thinking about like scientific machine learning, things like physics-informed neural networks, how that gets fed in there. So yeah, I think cardiovascular, I mean, we're moving towards it, but I think we have the same issue of everyone does it slightly differently.
I think it's interesting that you bring it up. We currently have a project here at the eScience Center, which is about physics-informed neural networks. And that's kind of the other way to do it, right? You can take an existing coupled simulation and add the neural network in somewhere as a component with a surrogate model, or it could be Kriging or some other method. And the other way around is to start with a neural network and then start putting physics-informed layers inside of it. For the former, using muscle is definitely, that works. That's essentially what we developed Muscle 3 partially for, and that's what we've done. For the neural networks, the tricky thing is that you are typically no longer time stepping. It depends a bit. You can put your neural network inside of your time stepping operator. But if you have a neural network that just tries to estimate the entire history in one go, then it becomes a bit more difficult to do that with muscle and MMSF because they're so tied to time stepping essentially. So I think at that point you may have to switch to different technology.
I don't know. All of the things we're doing with ML now are doing time stepper surrogates. In fact, at the moment, we're doing exactly what Roland did back in his postdoc with me, which was the vascular development simulations. We're surrogating those with time steppers. And perhaps unsurprisingly, they work extremely well. And they're very fast. So they work better than I would have expected. I expected certain kinds of errors to show up, and they do better than my guess was to be. So that's an interesting. I also want to appreciate coming back to the very beginning of your talk, where you use the Earth as a model. And all of the issues, of course, perhaps intentionally, all of the issues you brought up as motivators for these macro simulations of the Earth are perfect equivalent motivators if you replaced, medical digital twin of individual with digital twin with model of the Earth. Each of the things that you brought up, the lack of controls, the inability to make complete measurements, the stochasticity, the emergence, are all similar problems. And the fact that you can't do an infinitely detailed model at any scale. And so I think the analogies in the medical area are very clear. And so
yep.
I think it's a very compelling story.
It's the reason I mentioned the Earth is that this is something that I first encountered when I worked in geoecology, which is geoecology and biogeography are sort of a mix of physical geography and ecology from different perspectives. I worked with a professor in Amsterdam called Willem Bouten. He's retired now, but he was working on this, and he was a soil scientist originally, then a hydrologist, and then he became an ornithologist. So quite an interesting career path there. But in hydrology, they do quite a lot of modeling, and they look at these model identification issues. And he got interested sort of in these methodologies and in the fundamental way of how we do science and how you can do that with models. So that's why I got the idea, and that's why I always think of the Earth system first. But yes, a human is very much the same thing. We have another project at the eScience Center that is starting at the moment with most of the top ecologists in the Netherlands, and they're looking to make digital twins of ecosystems, which is, I think, on par with the human body in terms of the complexity of the system and also how little we still know about them. So that's a very interesting challenge as well. And yeah, it's similar. I think also simulations are going to be needed there because you can't just copy in a particular natural area. Sometimes you can, like they do experiments in the field where they take, for example, a particular patch of heath and they leave it unchanged, and then they go a few meters off to the side and they put a greenhouse over it and try to estimate what climate change will do because the temperature is higher. So at a small scale, you can do it. But if you're looking at larger systems, then yeah, you're going to just run into the lack of repeatability or the lack of control. Actually, during the pandemic, we had a similar thing with the whole masking debate. I believe at some point there were two adjacent counties somewhere in the US which were virtually identical in all aspects, except that in one case they were masking and in the other they weren't. And somebody made an estimate based on that, but I think that's still the only kind of natural experiment that we had. And so we still don't know whether masking works in a COVID pandemic. There were some simulations also, which I've seen in the press taken as like they took the assumptions of the model as proof that something had happened. It was somewhat unfortunate, but yeah, there are many things we don't know.
Okay. Any last questions or comments for our speaker today? I think we had a good discussion. Well, I want to thank you again. I'm looking forward to seeing how this develops, and I hope that you'll be getting perhaps some inquiries from our community, because I think this kind of framework could be very valuable to many of us. Thank you again so much for staying up this evening and talking to us.
Thank you. And I would love to collaborate, so I'd love to hear from you.